On our YouTube channel, you'll find a limited selection of pathology and patient videos. With Osmosis Prime, you'll get access to over 700 videos, including complete coverage of pathology and physiology, and a growing collection of pharmacology and clinical reasoning topics. Try it free today. Having a hypersensitivity means that someone's immune system has reacted to something in a way that ends up damaging them, as opposed to protecting them. There are 4 different hypersensitivities, and the third type, or type 3 hypersensitivity reaction, happens when antigen-antibody complexes deposit in blood vessel walls, causing inflammation and tissue damage. Alright, so first off, type 3 hypersensitivity reactions are mediated by immune complexes. Immune complexes, aka antigen-antibody complexes, are made of 2 parts, the antigen and the antibody. Antibodies, sometimes called immunoglobulins, are produced by plasma cells, which are basically fully matured and differentiated B cells. Initially, these cells make IgM, which can be secreted or bound to the plasma cell surface where it acts as a B cell receptor. When a B cell undergoes cross-linking of 2 surface-bound IgMs, it then takes up the antigen and presents a piece of it to T helper cells via T cell receptor to the MHC class II molecule presenting the piece of the antigen, along with co-stimulatory molecule CD4. The B cell CD40 also binds to the T cell CD40 ligand, and then the T cell releases cytokines, which result in B cell activation and class switching, or isotype switching, where it changes the type of antibodies it makes. In type 3 hypersensitivity reactions, typically B cells switch from making IgM to making IgG antibodies. Now remember that all antibodies are specific, right? Meaning that they recognize specific molecules called antigens, the second part of immune complexes. Antigens can come in all sorts of flavors. Some float around in the blood by themselves and are soluble, but some are bound to cell surfaces. Immune complexes are formed when antibodies bind to soluble antigens. Antibodies can also target antigens on cell surfaces, but these aren't considered immune complexes. And this is the first major distinction between type 2 hypersensitivity reactions, which involve antibodies binding to antigens on cell surfaces, and type 3 hypersensitivity reactions, which involves immune complexes with soluble antigens. A good example of a type 3 hypersensitivity is the autoimmune disease systemic lupus erythematosus, also just called lupus. In lupus, the IgG antibodies are typically specific for DNA and nucleoproteins, both of which are part of our own cells, making them self-reactive. Normally, your body should only react to things that are foreign, or not self. And this is maintained by a process known as tolerance, where only non-self-reactive B and T cells are allowed to mature, whereas self-reactive B and T cells aren't. This process, though, isn't perfect, and sometimes some self-reactive cells can escape, and these can mount an immune response against autoantigens, or self-antigens. With lupus, a DNA autoantigen might get released from a damaged cell, and a circulating self-B cell might find it and bind to it. If a T helper cell that's also specific for the same DNA autoantigen is close by, it'll help activate the B cell and enable it to differentiate into an IgG secreting machine, specific to that DNA autoantigen. Now what? Well, first off, there might be lots of this DNA autoantigen around, since DNA is in most human cells, right? Which allows a lot of IgG DNA autoantigen complexes to form. Now, if this were an infection, there'd be a lot of antibodies surrounding a large single microbe, which marks it for destruction by macrophages and other phagocytes. But in this case, the antibodies are trying to bind to a small, soluble antigen, and there might be lots of antigen relative to the number of antibodies. Small antigen-antibody complexes are less immunogenic, meaning they're less attractive to macrophages, and they don't get removed from the bloodstream as quickly. As a result, the immune complexes float around in the blood longer, and typically make their way into the basement membrane layer of various blood vessels. Once deposited, the immune complexes activate the complement system, a family of 9 small proteins called C1 through C9 that work in an enzymatic cascade to clear infections in various ways. In this case, C1 binds to the antibody-antigen complex, which sets off a chain reaction and activates C2 through C9. When a complement protein becomes activated, it's often cleaved or chopped by an enzyme, which results in little fragments. Fragments C3a, C4a, and C5a are anaphylatoxins and increase vascular permeability, meaning fluid leaks out more easily, which causes edema or fluid buildup. And this is the second major distinction between type 2 hypersensitivity, where complement proteins are activated in relatively small amounts, and type 3 hypersensitivity reactions, where complement proteins are rapidly consumed in large amounts, specifically C3 and C4, which means that the complement levels in the blood can be used to track disease progression over time. The second thing C3a, C4a, and C5a do is act as chemokines, meaning they recruit other cells like neutrophils to the site. Now, once neutrophils join the party, they try and phagocytize the immune complex, but usually they can't. During this process, they degranulate, meaning they dump a bunch of lysosomal enzymes and reactive oxygen species which cause inflammation and tissue necrosis, which ultimately causes vasculitis, or inflammation of the blood vessels. With inflammation comes further cellular destruction and more autoantigen release, repeating the cycle again. This most commonly takes place in areas like the kidney, where the blood's being filtered, causing glomerulonephritis, as well as the joints, since blood plasma is being filtered to produce synovial fluid, and this causes inflammation of the joints, or arthritis. And this brings up a third major distinction between type 2 hypersensitivity, where clinical symptoms correspond to the tissue where antibodies attach to and destroy cells, and type 3 hypersensitivity, where clinical symptoms correspond to the tissue where immune complexes are deposited, not where the immune complexes are made. Now, another example of a type 3 hypersensitivity reaction is serum sickness, which typically happens when a patient receives foreign serum and elicits an antibody response against those foreign antigens. A classic example would be to get bitten by a snake and then get serum with anti-venom antibodies. In response, the body would make some antibodies against the anti-venom antibodies. Now, if that person gets bitten by a snake again sometime later and gets the serum with anti-venom antibodies again, those antibodies that were made the first time around will bind up and make immune complexes with the anti-venom antibodies, which are now being treated like an antigen. These immune complexes then cause vasculitis and tissue necrosis, which would ultimately teach a person not to play with snakes.